כן, אבל לא
מה שעשתה?
כן,
זה קורס כאילו שלם שאפשר לעשות?
כן.
זה השנה שעברה?
זה סמסטר אחרון.
סמסטר?
כן. היה לו כאילו קורס על פרס נובל במדעי החיים.
כאילו, אני מתלבטת אם הם יהיו בגלל שהיה שלם, אז כאילו זה היה מעניין.
נראה לי שזה הרגיש אחריות על אחוז ערן מתעורר כזה.
גם חושבת שאני לא חושבת
יכול להיות זה תחום.
כמובן הדרך לנסגר
כן?
תלוי נראה לי.
לא, יש נראה לי שיש כאלה כאילו כשיש לך PA נגיד, אז לא אכפת להם. כאילו נגיד היה לנו קורס שכאן חרמון כאילו אחרי זה. היית איתנו פעם מולקולרית.
לא, הייתי מולקולרית.
היית מולקולרית. אז היה לנו כאילו ביום שני, אבל צריך קורס כאילו חרמון אחרי. אבל כאילו היא את רשומה אחרי. פשוט היה הקורס שלו. אבל אם אין PA אז פחות נראה לי.
לא, אם אין PA.
וכאן אין PA כאילו.
אבל אולי לפני
בכל מקרה זה הכל פילוסופיה עכשיו, כי כאילו הוא היה באמצע מצגת, הוא אמר שיש
אוקיי.
שיעורים מסתמיים טוב. לא, הוא אמר, כאילו, הוא אמר מראש, אני ממש צודקת את הדברים המנהלתיים הזה, הוא אמר מראש, שני שיעורים ראשונים, מבוא כללי, יחד איתי, הסתכלתי במצגת והוא הגיע כאילו ממש לאמצע. אני לא פעם נוגע, אני אחת לידה, לא רשומה.
כנראה ששינו את הזמן והוא לא היה מעודכן, שהוא התבלבל או משהו כזה.
Music nobody knows where it is.
No,
נראה לי שהוא כבר עולה. כבר תלמי לי בעד את משפט האלה. אתה לא כזה עדי, אתה בדרך למעלה שזה לא יעמוד.
Sorry, אמרו לי שזה I was told it was in 3 to 5, 3 p.m. to 5 p.m.
Sundays.
No, but for some reason it was in the last several years it was 12 to 2, Sunday and Wednesday. And this year they told me we want to shift you to 3 to 5. No problem, whatever you say. Apparently, only on Sundays. So I did not know that, and I have to tell all the other teachers because I told them from 3 to 5. So I apologize. And I hope you will forgive me. You will find mercy in your heart to forgive me. I just need to close all the things I was working on. And open today's lecture. Okay, so last time we were talking about how to quantify perceptions in animals, I believe. No? זה יעזור. open the... Okay, so this is the introductory lecture. I apologize for being late. This was not intended. And we were talking about some what is perception and the fact that it's not the actual stimulus that falls on the retina or on the cochlea or on the skin, etc. It's much more than that. When we talk about perception, when we talk about sensation, it's exactly about that. And this is much of what we're going to talk about in the course, about sensation, also a little bit about perception. And we'll go, these two topics will go hand by hand as we go along. Why is this not? No? Melach Mayim. Voila. Okay, we're here. So, and we talked about some of the history about this. Long time ago, people were talking about nature versus nurture. Is it all inside the brain, or is it all learned from a blank slate? And after that, some psychologists started to look at perception in a quantitative way. Okay, one way to look at quantification is to quantify decisions. Okay, and last time we talked about a ranking order, the number of times and each one has its advantages and disadvantages. Of course, as neurobiologists, we want to study the mechanisms in the brain, and for that, in many cases, we use animal models. And the question is, how do we ask an animal if it's, what is the perceptual decision it makes? And we can make animals, force them to make decisions. So, for example, we can have a go-no-go task where you present two stimuli to the animal, and it can report correctly, and that we call this decision a hit. It was correct. So let's say we give two stimuli, a blue and the red stimuli. We give it 100 times, okay, to the animal, and the animal has to report whether it was a red, it thinks it was red or a blue. We can, of course, do this in humans as well, so it doesn't matter for humans or animals in this instant. And then you have a 2x2 contingency matrix, where you get, if the stimulus is on the x-axis, we give it 100 times, for a total of 200 times, 100 times each stimulus, and this is the response of the animal, the human or the animal. And if it was a red, and the animal said it's a red, we call it a hit score. Okay? If it was blue and we said red, it was red and we said blue and we thought it was blue, it's a miss. Okay? If the stimulus was blue and we said it's a red, it's what we call a false positive. And if it was blue and we said blue, we call it a correct reject. Okay? Why do we say correct reject? Because for the animals, in most cases, let's say it's a go-no-go task. I think we looked at it last time. Yeah. Okay, so let's say you have, these are the two different stimuli, the red and the blue, and the animal is supposed to lick, to give a response only in the target tone, okay, and to avoid licking in the non-target tone. So if it avoids licking in the non-target tone, it's a correct reject. And if it's licking on the target tone, it's a hit. Otherwise, it was supposed to lick and it didn't, so it's a miss. And it licked when it was not supposed to. That's the worst thing. It's a false alarm.
What's the TO?
TO is timeout. It's a punishment that we give the animal in order to... Okay, and then we score the hit, miss, false alarm, and correct reject. This, of course, have to sum up to 100 trials that we gave these two animals, and then we can assume these are Gaussian probabilities, and we can plot the sensation that was evoked by the stimulus. Okay? So let's say this is red. This is the distribution of the red sensation. So if you're an animal, you have instances where the stimulus was very red for you. We would plot them here. Okay? And there were instances where it was, you were not sure if it's red or not red, but this was really the red. Okay? When we presented the red, it was here. On the other hand, we can plot the distribution, the Gaussian distribution of the other stimulus, the second stimulus. This is shown here on the blue. Okay? In many cases, it evoked blue sensation. Okay? So this is the sensation or the red sensation that was evoked by the stimulus. The higher it is on this axis.
It's kind of strange that though, because you wouldn't say that red and blue are on the same like.
So this is the sensation that the reddish sensation that this stimulus evoked as a red. Okay? Now let's say you did it 100% correctly. Okay? No problem. So these two distributions were completely separated. Okay? The red sensations were here. Sometimes it was very red, sometimes it's a little bit red, whatever. And the blue sensation would be here. Okay? And those were completely separated. You got 100% score, and the contingency table would be 100% here and 100% here. Great. But what happens in perception is that, certainly when we start to go to the just noticeable differences, these two sensations start to blur out, and you're not sure what exactly did you sense. So we can decide on a boundary area or a decision boundary in this plot. And if it was an ideal observer, this would be where these two graphs intersect in the middle. Okay? And this is exactly relevant to the psychometric curves that we showed here in the last trials. However, sometimes you have biases, and these biases cause you to shift away from the ideal observer boundary area. Now, the more the stimuli are closer together, the more overlap you have between the tables. And now you have to try and quantify to see what kind of perceptual quantifications can we make based on these types of probabilities. Okay? And one measure, and if you close the decision boundary right here, every time that you presented the blue and the animal said red, so it will be the false positives right here. Okay, if you cross the decision boundary here.
What's the axis?
The axis should be 1 normally. Okay, so all the 100% sensations, this is the number of events. Okay, in the y-axis, if you will, and this is the sensations that they evoked on the y-axis. The sensation are the x-axis. Okay, and there is a Gaussian. We think that, we plot these or we model these as a Gaussian distributions. So some of these evoke very strong responses, some of these very few responses. Okay, excuse me. Now we can plot a decision boundary, which is exactly here on the, shown here on the contingency table, okay? Every time, so this is 100% within this distribution, but every time we said yes when it was red, this is our decision boundary. Every time we said yes was this instance. So 65% of the times that we said yes were hits. But we also have a fair amount of false positives when it was red. Okay? So this would be the false positives would be here. And similarly for the other stimulus. This is the blue distribution of the second stimulus. And indeed, every time there was a blue and we said blue, which is on the left side of the decision boundary, we scored it as a correct reject, which is here. Okay? Great. And in an ideal observer, this would be the contingency table would be symmetrical, But normally it is not symmetrical, and this causes biases in the decisions, okay? And the shifts from the ideal region, if the decision boundary is on the ideal observer, it will be cut in the middle, and the mistakes and the correct would be symmetrical. But if we have biases, okay, let's say you have a serious incentive to say red all the time, and you don't care, you don't pay a price if you're making a mistake, no problem. This would be shifted to, the decision boundary would be shifted to a given side, okay? And the other way around. If you would pay a big price for making a mistake, the decision boundary would be shifted to the other side. But we can still quantify the distance between the distributions of these probabilities, which is basically plotting z-scores of all these distributions, plotting the distance between the average values of these distributions. And this distance is called d prime, okay? And it calculates quantitatively. Okay, sorry. So an ideal observer, like I said, has no bias. And in this, if you have a no bias, the hit rates and the correct reject would be symmetrical. And false positive increase. Okay, let's not explain that. And the measure, what I wanted to talk about, is a quantitative measure between these two different distributions is called d prime. And what's beautiful about d prime is that it is insensitive to the biases, because it really measures the distances between these two distributions. If the distribution is far away, like I said here, okay, if the blue and the red would be really far apart, there is no overlap, the d prime would be high. If you cannot distinguish between one and the second stimulus, let's say that they are on top of each other, okay, the red and the green distributions fall on top of each other completely, then the d prime will be 0. Normally we say that an animal that can differentiate between, so this would be a value of the sensitivity of the test, or how good you can differentiate between one stimulus and the other, the level of d prime. Normally, d prime levels of 1 or larger than 1 are considered that the animal can actually make the decision, or this test is good. And why I'm saying test is because historically, this probabilistic statistics came from the army, developments in the army. It was after the bombing of the Japanese of the Americans. And the army, the US Army, wanted the radar system of the American Army to detect the airplanes that are coming in, the Japanese airplanes that are coming in. Now, if you're a radar system, you want to know how certain are you that this is an airplane and not just a bird or something. What?
Nothing. A cloud.
A cloud.
Sometimes it happens.
That's a cloud, I see. So whether it's a cloud. If it's a cloud, you don't want to, that all your airplanes of the American Army will go look and attack this cloud. Now, of course, you can set the decision thresholds at different levels. So you can be super careful, and every cloud that you'll see, all the army goes in. But you don't want to. You want to avoid mistakes also. So then, all of this probabilistic statistics came, or the way to quantify a decision was based on these mathematical developments. Later on, it was used by a psychologist to quantify perceptions, as I'm showing here. And particularly this d prime is a value of the test that is measured, okay? How well the test is doing. These biases are a different thing. And the d prime is insensitive to the bias. In fact, this is widely used today in medicine. So for example, you want to know if you have a disease. You have cancer or you don't have cancer. If there is, they do some test, and definitely you have this cancer, you're on the right-hand side of the curve. But in many cases, the doctor, they have to make a decision whether it's red or blue, whether you have a disease or you don't have a disease. So exactly these measures are used a lot in biomedicine to evaluate the quality of the test, how good the test is, and this is evaluated by the D prime of the test. And there are almost no tests that you have full proof that these distributions are completely separated. Of course, in perception it is. Okay, if I give you a completely easy, no problem. Or if the test of the disease was super easy, no problem. You know, you do an MRI scan, you see the tumor, you know for 100% it's a... But normally this is not the case, and we live where things are blurry in the middle. So the origin of this was the US Army, engineers in the US Army. It was then adopted by psychologists to quantify behavior. We are still using it widely today. And then I think in the 1970s or so, it is also started to be used by the biomedicine community to evaluate the quality of the tests. Okay? So this is the D prime. Again, we are still in quantifications of perception, how to quantify perception. Yes? Yes, sure.
So when a biased observer does, it shifts the Gaussian.
No. What a biased observer does is it shifts the decision boundary.
Where is this decided, like what's in the graph says what's correct and what's not correct?
So this is the behavioral test, actually. Okay? So you gave the person a red, and he said either red or blue. So it's exactly these values in the contingency table.
How do I see in the graph what was the truth? I see here, see the yellow color?
So this is the truth, and this is... Okay? So if it was red and the person said yes, it's colored here in this distribution. If it was blue and the person said blue, it was here. Now, like I said, if it's easy and these are completely separated, the decision boundary can run in the middle and no problem. But normally, we want to study the limits of perception, etc., then these distributions they overlap. And this is true for the Japanese airplane that's coming in, or the evaluation of cancer of the patient. Okay? Normally there is a region where the sensations overlap. And you can't really decide perceptually what do you see or what do you hear, and they overlap. But we can quantify how good was the performance by the D prime, yes. The decision boundary according to this test. Yeah. No, the x-axis, I don't know the same. We are evaluating it from the perceptual test.
Because if there's no bias, you're expecting the decision boundary to be exactly in the middle between...
Here, to cut here, in the middle of the distribution. And this you can evaluate by the level of false positives to the false alarms and correct rejects. Okay, if it's symmetrical. In fact, because I was a little late, I will not go into it, and there are other more interesting things. A variation of this, to study this quantitatively, is the ROC analysis. Maybe many of you heard of, but that's basically a quantification of how sensitive your test is, and we can quantify from these parameters an receiver-operator characteristic analysis. Okay, and this is actually the name stems from these radar soldiers that came. You know how good they were in detecting whether it was good or bad. Okay, because basically that was their perceptual decision. And we want to have a quantification of the test in this case, or of the performance of the animal. Okay, because what we are doing is quantifying perceptions. Yeah. Yeah, fantastic. Okay, I'm going to talk about that. So this is exactly what we're talking about. What are the types of quantifications that we can and do quantify behaviors? Okay. So one is this, which stems from this. These kinds of distributions, things like D prime. Okay, this is just an example from our laboratory. I showed you this mouse running around and licking, right? I showed it to you last time. And basically, these mice they live in this, we call it the edu-cage. So in this automated behavioral cage, and they start to work at night when we go home about 7 p.m., and they work during the night, and they do many, many trials. Okay, and we can quantify how much hit rates, false alarm, correct rejection, etc. We do these contingency tables, and we can calculate D primes. So if the two stimuli are further apart, let's say, for example, 50% octave apart, it's a very easy task to do. So the D prime values are about, on average, of 3. Here you can see the behavior of a mouse, a single mouse. When it was 50% octave, this is the percent of correct responses, both hit rates and correct rejections, and this is the percent of mistakes he made. Okay, so in the contingency table, the numbers will be very high in the hit and correct reject, and will be very low in the misses and the false alarms. Okay, so you can do, he's doing very well. 90% correct, and getting it incorrect very low. Numbers, the D prime is very high. But then we make the test more difficult by pushing the frequencies closer apart. Okay, so basically what we do is we shift the distributions closer apart. Initially, he did almost 100%. And let's say it's an ideal observer. I don't know exactly, but I mean, I can know, but so he got it right all the time, no problems. Now we make the test harder and harder. Okay, and as you see that the D prime goes down until, above 1 we say the animal learns and differentiate, until he collapses altogether. And he gets it completely wrong. So the number of rights, it's chance level. The number of right versus wrong collapse completely. So this is one way to quantify the quality of the performance, the perceptual performance of an animal. Yeah, you had a question? No. Okay?
Basically allows you to say what is the
Performance of the animal.
yeah, but also
Fantastic. So what is the just noticeable difference of this animal? So when the animal collapses, you say he can no longer differentiate between 10 kilohertz to 9.97. Okay, in this case, it's a 10% of an octave.
Exactly when the D is under 1?
What?
That happens when the D prime is under 1?
No. So basically, above 0, the animal can still you say differentiate something, but basically, so that's what we call the criteria for learning normally. Because otherwise it's really within the noise level. And here you can see very clearly that the animal collapses. Okay? Now it's not doing 50% chance. Okay? You see that it's biased. Whatever. The animal doesn't care. Okay? He puts his neck in and he licks if it's right or if it's wrong here. He simply cannot differentiate. Yeah. So whether this is symmetrical? I didn't understand. What's the scenario that you are thinking of?
I think you're just mixing, in the experiment that you described now, there's no color, there's a tone. So if you can't detect the tone, you can be biased.
No, it can be a red and blue. I can ask the animal whether this is red or this is blue. Okay? And you will always tell me.
There is some specific, it can be a strike, it can be color, there is some specific stimuli that the animal should react to. So if
No, it's reacting, but it's making a mistake.
Like maybe he perceived it, but still didn't act. Like maybe that's
maybe the
Yeah, well, this is, again, these are all these biases that are put in, okay? I would expect if it was a mistake, the animal would make mistakes 50% of the time, or lick 50% of the time, and not lick the other way around. But what we see here is that the animal just licks to get the reward, and there is a bias towards licking, okay? In many cases, we see these fall down here. They're so afraid from the punishment that they get that they will avoid licking. And that's exactly the biases I talked about earlier. But normally, you would, if it's an ideal situation, it would be flat at 50%. And the decisions are pure in that sense. Okay. More questions? Maybe we can talk in that. D prime are unitless, actually, and it's taken from z-score tables of the different probability distributions. Yeah.
You said that solid line is the true detection, right?
Solid line is true detection.
The other line.
It's hit and correct reject. And the other ones are the mistakes, yes.
The sum should be 100 all the time, but the fourth bar, like, some of them is not 100.
So these are averages across trials, and it has to sum. Yeah. It's a smooth graph. This is for one animal. This is for 12 animals. Okay, and you can see that they start at around 50%, very fast. Once they get the punishment, very fast. They make no mistake, but they're still hesitant to live, to make a right choice. But then they're no longer afraid, so to speak, and they make choices and non-target. This is for the first step. Okay, but let's not go into the experiments we do here. We're just talking about quantifying behaviors. Yes, and time, yeah.
Why do you assume the variance is the same?
Excuse me?
Why do you assume the variance is the same? Maybe it's different.
Why do I assume the variance is the same?
The variance of
The Gaussian.
you have red and blue, so calculate it.
Yeah, so here this is the assumption which is based on these statistical model, and you're right. In many cases, they're not. The variance is not the same, and this is not taken into account here. Okay? So these peaks can be different, okay? Now I showed you the easiest solution. What you're saying is that, it can be distributions, the two distributions can be like that.
And
Yeah.
then how can you quantify the variance through an experiment?
So I don't know how we can quantify the variance here. In particular, this example, again, so I don't know how to quantify that exactly. But in this particular case, we're assuming in this model that these are Gaussian distributions. Okay. So another way to quantify a percept is the speed that the animal reacts. Okay? So let's say we give you a harder task. We gave you 50% octave apart, 2 tones, very easy, and we can measure how fast the animal reacted. Okay, I'll tell you, is this red or blue? You can score, is it red or blue? And in most cases, a very sensitive measurement of perception is reaction time. So in the easy tasks, the animal will react fast, and if it's a hard task, you will need more time to accumulate evidence for the task itself. Okay? And I did not bring it here, but within this task also, we quantify the detection time of the animal to make a decision here, here, and here, and indeed the reaction times goes up. Okay, which is another measurement that indeed these are more difficult tasks. This is again a measurement of, in this case, an odor sampling, not 2 sounds. You ask the mouse to differentiate between 2 different odors, let's say A and B, and that's the blue curve. Okay, and this is the accuracy of the performance of this mouse, okay? And you see that the accuracy is fairly good at, 100 milliseconds or something like that. Doesn't really matter. And the accuracy of this animal goes very high to differentiate between 2 different stimuli. And now I can ask him to differentiate between harder and harder and harder tasks. And what you see is two things. One is that the accuracy falls down. It is no longer 100%. This is the type of things that we talked about when we looked at the contingency table. But also there is a shift to the right of the table, and it takes, for the same percent of accuracy of the animal, it takes them more time to sample to get the same level of accuracy. And this is called the speed-accuracy trade-off. Okay? If you have to make a faster decision, you're less accurate. And this is a whole big field, very interesting field of decision-making and accumulation of evidence, and how does the neural system does that, etc., etc. Okay? We will not get much into it. This is ABC of sensory physiology. But there are ways to measure quantitatively perceptions. One is D primes, for example, ROC, which we didn't go into, which is a variation of the D prime, and another one is speed-accuracy trade-off. Okay, so let's look at an example of how far response times can take us. So this is an example of a psychophysical experiment of humans done by Ant Riesman, which actually had a huge impact on the field, on the search strategy of an animal, in this case humans. And what she detected here is she gave the observers to find, in this case, the blue cross in the image. Okay? Great. And you can see this is a very easy task. You can find the blue cross in the image very fast. And what she did is she made the array of this block bigger and bigger and bigger and bigger, and still the response times of this is a 5 by 5 matrix, okay, and the blue cross was here, and she asked how much time it takes the animal to quantify the blue cross, and if it was 5, 10, 15, 20, or 25, it didn't matter. For the humans, it was very easy to identify the blue cross because it was very different than its immediate behavior. When you ask a more difficult task of the animals to identify the blue cross over this matrix, okay, or even harder to identify the blue cross when there are mixes of colors and greens, you can see a very clear increase in detection times. Moreover, if you ask the person not to identify the red cross in the matrix, but to say whether it is missing in the matrix, it will take the person double the time to the time that it takes them to say whether it is there or is it not there. And you do it thousands and thousands of times. On average, sometimes the cross will be here, and the person will say, yeah, I hit it here. So on average, it will take the human half time to detect the cross, whether it's absent, because then it has to go through the whole matrix. And the meaning of this is that this kind of a task is what she called a serial task, when she went line by line, when the person has to go line by line to detect the feature in this image. While here, it's a completely different task, okay, of a parallel search to see whether there is a different object in the field. And it took him less time. The same people takes less time. And still, if you say if it's absence or is it not absent, in this kind of a field, it doesn't change the time. Okay, so the task is not different. And this led to a full theory that object recognition like this, okay, so if you just want to see if there is something out there, is something that is done faster in the hierarchy. Okay, and because simple things, and we will talk about that when we will talk about sensory information, initially in the nervous system, the stimulus is broken down to its basic features, okay? So wavelengths, colors, frequencies, etc., etc., okay? And the percepts of an object, higher order objects, are considered to be the responses further along the hierarchy. If you remember last lesson, we saw the Simpson neuron. You cannot find the Simpson neuron in the cochlea or in the retina. Okay? You will find the Simpson neuron higher along the hierarchy. And this led actually to an influential theory, which was actually started by Merav Achisar and Shaul Hochstein, which is called the reverse hierarchy. So if you go along the brain stations, you will find neurons, and also we will talk about the Hubel and Wiesel experiments where we see neurons responding to very basic features of the stimulus. But if you stick your electrode higher along the hierarchy, you will see more complex and complex features. What they claim is that the perceptual decisions is actually done first, higher here. Okay, so it's easier for you to find the different object, the cross, within an array of red and blue and green crosses, higher along, and you can make the decision. However, if you need the details of the decision, you have to go back by feedback connections to sort out the details that are early along the hierarchy. Okay? So this flips a little bit of what we think that everything is feed forward. Very early on in the nervous system, things to start to feedback, and loops start to become very, very complex. Okay, so this is all I wanted to say until now, up to now, about psychophysics, which is the relationship about the stimulus and the sensory experience. This was early on in history. Okay, psychologists made impressive advancement of how we understand psychology and perceptual decisions. And only from 1950s or so, we started to sink our teeth deeper into the sensory physiology, okay, with the inventions of electrophysiology and other tools. And in the next, we're going to take a, let's say, 10-minute break until 1:05, okay? I apologize, I owe you your 15 minutes. It was not intentional, and we'll talk about sensory physiology, and by this we will close the introductory lecture. Okay, any questions until now? Let's take 10. I'll drink a cup of water and yeah, sorry. לניסויים האחרונים? כן. זה. פה אני עושה מה שנקרא corrected ratio. במקרה שלו הוא צריך לא ללקט, הוא לא לעשות. באזורים שונים, אוקיי? אז הביצועים הם שונים, בהתאם לסטימולוס עצמו.
כן,
אז זה לא אומר שאת לא יכולה למדוד את היכולת שלו להפריד בין הירוק בין הסטימולוס הספציפי הזה לסטימולוס הספציפי הזה. את אומרת לי, אתה לא יכול to generalize ולהגיד, זה היכולות שלו. כי אם תשתמש בכחול, אז יכול להיות שיהיה ביצועים אחרים.
אני אומרת שאם תעשו את זה, עדיין, הוא ירצה...
את רוצה רק לעשות ריברסה?
כן,
הבנתי, שיהיו אותם ביצועים.
אז זהו, אם הוא הופך את זה, אז אתה יכול לומר שבמידה, אבל אם לא, אז בעצם בפעולה עצמית, הוא מבצע את אותה פעולה עם הירוק ירוק.
אפשר לעשות את ההיפוכים ולראות אם הדבר הזה הוא סימטרי ומסכים איתך להחלטה של קרנות ולמדוד בה. אבל לא דיברנו על זה פה בכלל. about the reverse hierarchy thing. Okay, so normally people, the most common way to analyze the brain is bottom up or feed forward. Okay, so initially the computation is done in the cochlea to break down the frequencies to different frequencies. Then later on in the next station we combine the different frequencies into a new concept, and then we build until we realize that this was Simpson. Okay, Simpson cannot be done in the cochlea at all, and therefore it's about building up the information. So there's no question that the information is feed forward. Okay, these neurons synapse here and synapse here and synapse here, but there are many loops and feedback loops that go along. What Merav and Chaim and Shaul did, it says that decisions, perceptual decisions, are first made high in the hierarchy. Okay, and this they could know by the parallel searches. Okay, so it doesn't matter. It depends. If you need to look on the details, and the perceptual decision is harder, you need to go back to the earlier stations. But in the context of this talk, it doesn't really matter. It matters only about quantifications of perceptions, and we talked about D prime, etc. And then I talked about speed measurements, okay, which is a very common way to quantify persons. Okay, how fast the animal made the decision, and this has impact not only about how hard the task is, like I showed you, but also, again, just a teaser that it has. People can use this information for deeper thoughts about perception. Okay, and the second part of the talk today has to do with the sensory physiology. But before we go down that road, I would like to ask you, what would you like to know about? And I think I asked you this last time. What do we want to know about the system that we want to study? Now, let's say you make a decision, I want to study this part of the brain. And I want to understand, so Mao told you want to understand what is the problem, what is the algorithm that would solve this problem, and how does it do it, okay? And we're in the how does it do it part. So what do you want to know about the brain circuit that you're going to analyze? Okay, this is a black box. Now we want to understand the mechanism of how does it do it, whatever it does. So what are the things you want to know? Yeah?
I want to know the input and output.
Input and output are critical. Okay, what is the input to the system? What is the output to the system? That's critical. What else do you want to know about your black box? What is the basic unit of computation? Fantastic. Is it a neuron, a group of neurons, things like that?
How big is the black box?
How many neurons I have in my box? What is the connectivity within the box? What is the spiking patterns of each neuron in the box, etc., etc. Okay. So all these details we want to know. We still want to know today. It's 2016, and if you take almost any neural network besides the very peripheral sensors of the receptors, which we know quite a bit a lot, once you make a step or two synapses into the nervous system, we still don't have a good understanding of the inputs and outputs to the system, the cellular physiology of these cells, how fast they can spike, how many spikes per the input, which is basically the input-output, the connectivities, the strength, the sign, the inhibitory, excitatory connectivity within all the computation units, the different cell types you have, where they project their axons to, etc., etc., etc. So all of this is about to understand the mechanism of how does the brain do whatever it does. And I like to make this analogy. It's actually not me. I stole this from a friend from genetics. Okay, which he said, okay, we can make analogies, but let's not insult the neuroscientists. Maybe I should close this. So I won't be on tape. Okay, but we are really at the point, as a biologist, I see how when we understand some processes of here in this table is to make some kind of analogies, not exactly 100% alike, but some analogies between what we know about genetics and cell biology to neuronal circuits. So we want to understand the building blocks in genetics and cell biology, or genetics. It's a gene. Here we have about 20,000 genes in our genomes. Great, no problem. In the brain, it's the single neuronal computation. In many cases, it's a neuron or a group of cells within the brain circuits. We have billions in totals, many, many types of different cell types, etc. So it's getting a little bit more complicated than genetics. Maybe, don't know, because there are all kinds of interactions there. Information processing would be equivalent to proteins interactions, things like transcription, translation, degradations, etc. The information processing of neuronal circuits is the neuronal activity, again, spike trains, excitation, inhibition, etc. And the experiment as experimentalists, many of you won't be experimentalists, but the ones who do, the best, there is a very simple and straightforward way of saying whether something is important or not. In genetics and cell biology, it's gain of function experiments and loss of function experiments. You do rescue experiments, and you can understand whether something is necessary or sufficient for a process that you study. In neuronal circuits, it was hard, and we used to do pharmacology, lesions, but there are all kinds of immediate feedback loops that make this thing harder. The revolution of molecular genetics and optogenetics into our toolbox of analysis allows us in a much more elegant way to stop networks of working and activating back, or gain of function into the system, etc., etc. Okay? So you are joining neuroscience in a very good time in history, where these tools are ramping up. And I don't want to sound old, because I'm young, still young, etc. But the type of experiments that I could even dream of doing, even not dream of doing, when I was starting my PhD, it's a completely different ballgame now. So the tools that we have today are, weren't even in my imagination when I started. Because you come as a student, this is the arsenal of things that you can do. And this is really an evolving field of study, neuroscience. So you're making the right decision to go into the right area of research, and the tools are cooking to be better and better. Okay? Okay, so a few general issues about sensory physiology. First, sensory information is gated into the central nervous system by stimulation of receptor cells. Thus, receptor cells provide the first neural representation of the external world. This we understand quite well. Okay? In all sensory systems, we understand fairly well how receptors interact with the external world. We will learn about that. As we go along each sensory system, we will talk about that. But then information flows, as I said, in serial streams, parallel streams, feedback streams along the way, and information is continuously transformed. Sensory pathways are recursive, and this is far, far, far less understood. And this is where you will find yourself doing neuroscience, okay, mostly. Or you can, of course, deepen our understanding of the receptor information processing. But we can say fairly well that this is best understood, this is less understood. This is also true for single neurons versus local brain networks. We understand fairly well the spike generation, Hodgkin-Huxley equation, etc. If you have heard about it, the biophysics of the cell, fairly well. You go into 2, 3, 4, 100, 10,000, 500,000 neurons, we know much, much less. Okay. General principles. Those of you who took physiology know this slide. Basically, The brain is not a syncytium, not all neurons are alike, different neurons do different things. This is obvious. Okay? Okay, the sensory epithelium. We're going to talk again mainly about the five senses. In vision, it's the retina where photons start their journey. In audition, it's the basilar membrane and the organ of Corti, which you will learn about again at the end of the course. All of this will be your bread and butter. Touch is about receptors on the skin, smell, receptors in the olfactory epithelium, and taste in the tongue. I don't know if we will have enough time to go to taste. It's actually super cool, a super cool sense, very simple, but I'm not sure we'll have time. Maybe not. Okay, we'll do mostly other readings. Okay, this is the hair cells of the cochlea. This is the photoreceptors on the receptors there. Yeah, so each one is a different concept, different thing. Okay, so the cochlea is a one-dimensional array of hair cells, and they're sensitive to pressure waves, like we talked before. The retina is a two-dimensional matrix. It's sensitive to light. These neurons are sensitive to light. And at the fingertips, for example, these particular cells respond to pressure, but we also have other receptors that respond to chemicals, like in pain. And you will learn about that. Great. Okay. Changes in physics, in the physical variables that this type of an animal can sense, or any other type of animal sense in the brain, is transformed into electrical activity. Electrical activity is the language of the brain. And these transitions from the physics of the world, from pressure waves, electromagnetic waves, etc., into the brain, these potentials are called receptor potentials. Okay? And this is where we interface between the physics and biology, kiss each other and interface, and start their journey into the brain. These potentials evoked are called receptor potentials. And again, we know them fairly well. We understand how they arise fairly well. Question?
I didn't fully understand, what do we call receptor potential? I mean, we translate the information to electrical activity?
Yes.
It's called the receptor potential?
So the receptor potential is the electrical potential, the membrane potential changes of the receptor cells. Okay, and this is the first transformation, or this interaction is the first interaction between physics, the physical world, and the brain. Yeah.
The slide says that changes in physical variables cause these changes, but if we have a constant...
Yeah, that's fine, yeah.
So it still will cause electrical activity, or not?
We will see later on, okay? So normally it's the change that you... yeah, but this is continuously, it's a dynamic system all the time, okay? And some have potentials have spontaneous activity, and this changes, but yeah, normally we detect change. Normally it's the change that we are sensitive to. So there is like a basal condition, and we speak about, sensation when something has changed. Yeah.
Okay, so potential is the output of the first layer, right? The sensor layer.
Not yet the output. Even the membrane potential changes of a receptor cell. So let's take a photoreceptor cell, stick an electrode in, shine light, and you see a change or a hyperpolarization. Okay? So this is called the receptor potential. Great. Okay, and now there is an issue of payment, okay? We have to put our money where the transition is. So it's not the energy of the stimulus. The energy of the physical energy has, you can quantify how much energy it is, but it's not equivalent to the electrical energy, okay? What do I mean by that? Let's take, for example, phototransduction of a photoreceptor cell in the retina. Okay, we know by psychological experiments that we can detect single photons, and a single photon energy is equivalent to 10 to the minus 18 joules, whatever. Okay, it has a certain amount of energy. Great. Now we record from a photoreceptor he's happy, no problem. Membrane potential, I don't know, minus 60, whatever. And then we shine a single photon onto the cell and we get some kind of membrane potential. Okay? Normally a single photon will cause a depolarization or a hyperpolarization of a few millivolts. Okay? A few millivolts is worth about 10 to the 4th ions of movement from one side of the membrane to the next. But eventually, the energy that could make this potential is paid off by proteins on the membranes of the cells. In this case, if you are a biologist and you took Introduction to Physiology, you know this guy very well, the NAK pump, okay? And it requires each movement of this pump. This is an electrogenic pump. It transfers 2 ions of potassium versus 1 ion of sodium, and it allows the different concentrations of ions in different sides of the membrane. So for each ion that you have to keep different in a different concentration, you need 1 movement of this pump. And we know that each movement of the pump, you have to pay in energy 1 ATP, 1 molecule of ATP, molecule, okay? The energy of a single ATP molecule is about 10 to the minus 18 joules, roughly. Okay, this means that in this particular case, where you need 10 to the 4th ions to get this depolarization, you pay 10 to the 4th more energy than the photon to start. Okay? So, and the transition is a biological transition. So this is physics. It has a certain energy, this photon, and this energy is transformed into electrical activity, which is the language of the brain. And these are normally paid off. It's not the same energy, okay? But there is a huge gap or a huge price that we pay in energy in order to do this simple transformation at the receptor potential. Yeah?
So it's essentially, it's not analog, it's not digital, it's not analog.
It's what?
It's digital, it's not analog. Because it's not exactly digital.
Not exactly digital, or sampling. It has some aspects of digitization. We will talk about that soon. But no, what I'm trying to say is that there is a transformation in the receptor cells from physical energy to electrical activity, to spice of the cells, whose energy source is metabolic. And this process is called stimulus transduction. It's a biological process. It's when physics interfaces with biology. But basically, there are now cellular cascades, which protein detects the photon, and what is the conformational change. And this in turn causes different molecules to open, put channels on the membrane, etc., etc. Okay? So we're starting our journey. We're starting from the physical variables that we want to detect, and we're translating them into electrical activity. Different senses do it in a very different way. Okay? There is direct mechanical coupling, for example. Okay? This is done in somatosensation and hearing. You will see that the pressure waves actually deflect the hair cells in the cochlea. So this is a mechanical issue. In some cases, it's ligand-gated channels. Okay? A chemical will join a protein which is on the surface of the membrane, and this will cause the electrical changes. In other cases, it's second messenger cascades. Okay? Different molecules that you start with at the membrane will cause all kinds of cascades, like in vision and smell, that induce the electrical changes in the receptor cells.
Don't most of them happen, like all senses happen by some sort of cascade?
No, sometimes it's the channel itself that sits on the membrane, and it's hit on the channel, and these are open. There are channels that open by direct pressure, and each sense is a completely different story. This course is about the details of these different senses. Okay, we want to know how do we hear, how do we smell, etc., from the receptor surface up to perception, up to psychophysics, and hopefully you will get, by the end of this course, you will get some of the flavor and the tools that people use to quantify and analyze and study of what we know and what we don't know about different senses. Okay, that's the idea of this course, basically. Okay, so there are some tables in the textbook that shows you, what is the energy, what is the receptor cell that detects it, etc., etc. Okay. Spike generation, yes. On the surface you get receptors, potentials. Not always you get spikes from the receptor cells. In many cases, the receptor cell makes a synapse onto another neuron that only then has an action potential, and normally these are involved with sending the axons into the central nervous system. Okay? So from receptor potentials, we're then moving to spike generation. Sometimes it's within the same cells, in many cases it's not. It's downstream, and we'll talk about the different details there. Okay. Neurons. Neurons are both spatial as well as temporal filters. We'll talk about that and basically finish the talk today. Hopefully I'll manage to finish on time, and if not, maybe we'll do a few minutes on Sunday when we talk about the paper. Okay, and I just want to give you some basic, and again I apologize if people, some part of the people here did the ROC for their master's program or whatever, and doing electrical encodings, patching neurons is your bread and butter. So what I'm talking here is very basic. So we have to align. Okay, so in many cases, receptor cells and neurons in general have receptive fields. And one can quantify, for example, I'm studying photoreceptors. We have 3 types of cones and one type which is called rod. You will learn about that. And they are sensitive to different wavelengths of light. So we can quantify, in this case, how many spikes these different neurons gave when I shone light at a given intensity at different wavelengths. Each neuron will have some kind of receptive field that you can quantify. Okay? So one can characterize a neuron by the width of its tuning curve. Okay, so how wide it is. It can be narrow tuning, it can be wide tuning. A neuron can be redundant in a way that it just responds to a photon of light, whatever it is, or it can only respond to a specific wavelength in this case. And you can quantify the best frequency of the cell. So cones, red cones, have a best frequency at about 564, green at 533, blue cones at 437, etc. Of course, then a problem arises because you cannot extract this information of color based on the spiking activity of a single photoreceptor, right? Because if it has a tuning width, if it's sharp on, no problem. But if it has a tuning width, he will spike similarly at 500 nanometer and at, 620. So you cannot really make sense of what color it was in the world if you study only a single neuron. Here is where neuroscience starts to become interesting, because you have to now compute what kind of information was there based on a few activity of a few cells together. Okay? Yeah.
If I have 2 stimuli for the same neuron,
Yeah.
I can identify the location of it.
The location.
Different 2 points.
Yeah. True. No, this is an easy problem. Yes.
I didn't understand why the signal neuron
So if you ask the neuron how many spikes you're recording from a single neuron, okay? Let's look from the side of a single neuron. If you give him 500 nanometer, he will spike this much. Give you more, he will spike this much. This is the tuning curve of the neuron. Maximum number of spikes at 464. I'm just saying that the same, because it has a tuning width, the same neuron will give you the same number of spikes for different stimuli. And it's very hard. Now you can play the trick and now change the wavelength like, what's your name? Alex suggested, but generally speaking what I'm saying is that you need to do some kind of games in order or to detect from many neurons or to play different frequencies. Normally a single neuron will not be enough, although we saw an example where it is, okay? The Simpson neuron was very clear, even if you give them just a recall, it was clear. Okay, some more measurements or expressions you need to know. A raster plot is when you see the stimulus, the spiking activity and the time. X-axis is time, y-axis is stimulus in this case. The stimulus, the different frequencies, and in this case it was given here. So you see this neuron spikes. This is auditory frequencies from our laboratory, from 3 to 80 kilohertz. You see that this neuron has a receptor field somewhere in the middle, okay, in the mid-range of these frequencies. Other expressions are what you would call a peristimulus time histogram, or PSTH in short, where basically you take this and you collapse it on one projection, and you see that this neuron responded strongly at this time, or it can be an off response or a late response, and we will start to see similar things as we here it's a spike rate, okay? So how many spikes per time bin?
But in the last plot, what shows the time being?
No, this is time from 0 to, 600 milliseconds, for example. The stimulus was given here, and you can see that the neuron responds with such and such spikes during this time window. But neurons vary a lot, okay? They can respond wide, they can respond with off responses, multiple peaks. It's, this is a very basic, simple neuron. Just the main expressions. Okay, the way we build this, we build, for example, in audition, we build what we call an FRA, a frequency response area. So you can see when we will talk about audition, you can use a high intensity stimulus, and this is the PSTH that you get. This is the raster plot that you get, okay? And you see this neuron responds to low frequencies. Another way to show this is to now flip the axis of the frequency to this axis. Okay, so now it's from 3 to 80. Before it was 3 to 80 here. And you can see and just plot the color code of the spiking intensity, like in the raster plot above, and you see that this neuron responds to high frequencies to high intensity or high spike rates at low frequencies, okay, between 5 kilohertz or something, 6 kilohertz, that's the maximum. But then we can do the same thing when we decrease the intensity of the sound, okay, so now let's decrease and see the PSTH and the raster. So you see now he still responds here with a lower intensity. It's now 56 dBSPL. We'll talk what dBSPL is when we get to audition. And you see the raster plot on the top. Okay, so let's go back. That's the high intensity. Look at the raster and the PSTH. This is a lower intensity. Now we do a lower intensity and lower intensity where now this neuron does not respond at all. And this way we can build a tuning curve, so to speak, of this cell. Okay, great. And like I told you, normally information is quantified. Information is quantified, or computations are done in many cases by populations of neurons and not single cells. Again, just to give you a flavor of how real neuron from real brains look like, this is a neuron that we record in our lab from the auditory cortex, responding to a song. Okay, you see the pressure wave here. This is actually a baby song of a pup calling its mother. And you see each block is a neuron, and this, what you see here are the raster plots. Okay, so you see this neuron number 14 responded with and each one is a trial, different trial. You see that this neuron responded strongly when the noise began. This neuron responded to each and every syllable here. This neuron responded only to before syllable number 3 and syllable number, 12. And you see that this neuron responded only initially and then was completely quiet. So basically when you want to decode a network, this is what you have to deal with. Okay? Very different neurons do very different things. We know very little about the neurons. Sometimes we have information about their spike rates, if it's simultaneous or not simultaneous. All of this has a very important impact of the sensory code. Yeah.
Would you say that, let's say, you hear very high frequency sound?
Yes.
And the sound is also more, so the volume is high up. So
Yeah.
there could be, the cell, one neuron spike for both different, like.
Yeah, so neurons can be tuned to frequency, neurons can be tuned to intensity. So you can have the multiple peaks within an FRA. So for example, what you were referring to is a neuron that would respond only to high frequencies and only to high intensities?
No, I mean.
Low intensity?
Four, not and.
Yeah, so you find different things. As you go higher into the brain, into the hierarchy, you find more and more complex receptor fields of the cells. And we'll actually see it when we talk about the paper on Sunday. Okay, about complex and simple cells, which were actually one of the first recordings ever done. And this brings us to our giants who got the Nobel Prize, Hubel and Wiesel. Actually, the first one was Vernon Mountcastle, studying the somatosensory system in monkeys and mapping the brain. But soon after, Hubel and Wiesel started doing experiments in the visual cortex of the cat, and we're going to talk about this paper. This paper is going to be in the exam as part of your curriculum, classical stuff. Okay. You don't see this. Sounds very good. Yeah, we're going to see this movie. This is the real live experiments of Hubel and Wiesel. So there's an electrode in the brain of the monkey. You're hearing the spikes, and what they're doing is they're taking this bar in the visual cortex, they're moving it along the screen, which is in front of the cat. And now they're marking by hand where is this neuron responding to. So you see, every time it's here, in this diagonal, it will respond. And this is the way they did experiments then, 1962. Now they make it larger, they are looking for inhibitory regions. You see now the neuron is inhibited, he's marking inhibitory region. You can hear the spikes. Yeah, the excitatory center, the surround inhibition. Great. So we will talk about these cells, okay? But this is how they did experiments then. And now the amazing thing is that when they tilt it, you'll see nothing. So it's the same receptor field in the visual field, but still it responds only to these tilted, these orientation bars. We'll talk about that on Sunday, okay? Let me find my... Okay. Great. And, let's look at a modern paper. Yeah. Okay, so if we look at modern, now we can do 2-photon imaging in virtual realities. I was late today, so we're not going to go into the full movie, but basically Oh, sorry. Sponsored by Gillette. Sorry, soon we will. It's just some pictures on YouTube that you can log in and. No? Hello? Sorry. Anyway, so these days you have these. So this is a mouse running on a wheel. His head is fixed, and now he can turn around, left and right, go up the stairs. Kill the bad guys, etc., etc. And we can put 2-photon microscopes on the brain of the other movies. You can put 2-photon microscopes when the animal makes a decision to run left or run right. Other issues is with the electrodes implanted into the brain, so animals can you can really record the spiking activity while the animal makes a decision and behaving. Okay, so this is 2009 or something. Yeah, so you can do, okay, thank you for the question. This was not an implanted question. You can do different types of recording. So first of all, the holy grail, the gold standard is electrophysiology. We have different methods to record from neurons, from single channels, to spiking activity, to synaptic currents, to local field potentials, EEG, etc. Each and every method has its own advantage. We can do imaging experiments. This is again an example from my lab, doing 2-photon imaging. What you see here is an image at the top of the brain, -50, an animal injected with a dye, and we can go down deep, change the focus of the brain with optics, go deep into the cortex, until we find a nice focal plane that we like, that we injected the cells with. Now we do it with genetically encoded dyes. So let's say you chose, you stop here, in a nice place, about 400 microns deep, and then you can record simultaneously, optically, in this case from 16 neurons, we can do hundreds of neurons now, simultaneously, and you can imagine that this is now a gold mine for computational neuroscientists, okay? We can ask not only whether this neuron, let's say there was a stimulus here, responded or not responded, but whether all of the population responded together, etc., etc. Yes?
In your video, there is a black dot in the middle that goes along all the...
Yeah, this is a blood vessel. So actually you can follow the blood vessel. This is a blood vessel, goes into, you can follow it deep into the brain and it branches, etc. These are blood vessels. The green are actually neurons and the red are glial cells. And they won't spike, of course. So there are microscopes that one can implant on rats like this and do full population imaging on animals. There are now newer technologies. This is pretty old. What time is it? Okay. Sometimes neural activity correlates very nicely with perceptual performance. So let's say I'm asking you to rank perceptually how intense was the pressure on your skin, okay? And you can do it pretty well, okay? As I increase the indentation of the skin,